Thank you, Dr. Singh. Good afternoon, everybody. I want to first thank the session organizers for including me in this important discussion. I am a biostatistician by training. Background for this session are listed here. We are here because of inadequate representation of older patients in cancer clinical trials, although there is rarely an upper age restriction specified in eligibility criteria. We know that there are many other reasons for this, and the question is, can we design clinical trials in a way that would increase participation of older adults? Without compromising safety, we can examine what are the available trial design options and how we can interpret the data from such a trial. Here are a few points to consider: prevalence of disease in the older population, access to care facility, frequency of outcome assessment, where the outcomes are invasive or, whether the outcomes are invasive or non-invasive, and more importantly, what are the essential data needed to evaluate the treatment under consideration. Thoughtful consideration for exclusion criteria should also be considered, such as the specific toxicity of the drug and any drug-drug interaction. We can include older patients in all phases of clinical trials. During the early phase, one could consider adding a small cohort of older patients to understand safety and tolerability of the treatment, dose selection, and potential dose modifications needed. As polypharmacy is common in older patients, study of drug-drug interaction and understanding feasibility of outcome assessments in the early phases are valuable. And all of these can guide in planning and designing confirmatory clinical trials. In the confirmatory phase of drug development, there are trial design options, and I will go over a couple of them in the next slide. Excuse me. Post-marketing studies may also be considered to understand safety and efficacy of the treatment in older patients. As detailed in the FDA guidance, here are a couple of confirmatory trial design options. The overall population can be split into a restricted subgroup of less than or equal to 75-year-old patients and a higher risk subgroup of greater than or equal to 75-year-old patients. Either this could be purely based on age or based on some comorbid conditions or other risk factors too. The patients can be stratified by age at randomization. The primary analysis can then be based on the modified ITT, intent to treat population, or MITT population of only the less than or equal to 75 years of age. A hierarchical testing procedure where hypothesis based on ITT is tested after testing in MITT population can be considered. And an adaptive feature to stop enrolling older patients based on interim analysis if safety concerns arise can also be included in the design of the trial. We can also simultaneously do two studies, a randomized study in less than 75-year-old patients and a single-arm cohort of greater than 75-year-old patients. The two in this case will be analyzed separately. However, only descriptive statistics can be obtained from the single-arm cohort, and we will not be able to interpret time-to-event data from single-arm studies. In conclusion of my talk, here are some key considerations in planning and designing a clinical trial. First thing is, who should be in this expanded older age, high-risk population? what comorbidities, drug-drug interactions, level of toxicity are acceptable and they can be included in the clinical trial. In the randomized controlled trial, what proportion of patients in the older age group should be included? One option is, of course, to include based on the proportion, the prevalence proportion of older patients, or should we limit number of patients in this high-risk stratum? Then when we are testing the primary hypothesis, we have to, of course, consider the type 1 and type 2 errors. And since most of the cancer trials have time to event as the primary endpoint, the analysis will be based on number of events. And so what are the total number of events for the final analysis so that we can make a benefit risk assessment, first based on the restricted population of less than 75 years, and then, of course, use the rest of the population of greater than 75 as well. We can do a hierarchical testing, but is this feasible is another question to be asked. What if more events occur in the high-risk stratum, then that will be contributing more to the total number of events that we expect for the final analysis. So we may want to restrict the number of patients who are over 75 to be included in the clinical trial. Consideration also of safety assessment in the high-risk population. Assuming that there are comorbidities in this group, then the approval of the product itself may be based on the less risk or the lower risk stratum of less than 75-year group. However, the safety that's evaluated in the greater than 75 years is also included in the product label so that the physicians can better manage their patients. The single-arm cohort of older age in the high-risk population is, of course, we can do this simultaneously or post-approval. But the advantage is you could consider enrolling patients only in certain sites, for example, where more geriatric patients are seen. But it will be difficult to interpret toxic events, in particular deaths, without a control arm in a single-arm study. Post-marketing studies are another option. However, we have to consider whether the outcome definition and assessment are reproducible. And measure of safety evaluation, consistency of recording the data, all of these are important. Thank you. And I can answer any questions.